the lymphatic system. So the lymphatic system consists of two semi-independent parts. First is the meandering network of lymphatic vessel and the various lymphoid tissue and organs scattered throughout the body. So this is an example of a capillary bed, arteriole, precapillary sphincters, capillaries, then the capillary network and the venule. You can also see here the flow of interstitial fluid into lymphatic capillaries, where it is called lymph. So this is the arteriole, small artery, then smooth muscle, endothelial cells, lymphatic capillary, blood capillaries, loose connective tissue, venule, so that is the interstitial fluid and the lymph flow. The lymphatic vessels transport fluids that have escaped from the blood. As blood circulates through the body, exchanges of nutrients, waste, and gases occur between the blood and the interstitial fluid. The function of the lymphatic vessels is to pick up excess tissue fluid, now called lymph. Lymph means clear water, and return it to the bloodstream. So the lymphatic vessel is also called lymphatics from one-way system, and lymph flows only toward the heart, going to the heart. Then the lymph capillaries, the microscopic blind-ended lymph capillaries, spiderweb between the tissue cells and blood capillaries in the loose connective tissues of the body, and absorb the leak fluid, primarily water and a small amount of dissolved proteins, are so remarkably permeable that they were once thought to be open at one end like a straw. The lymph is transported from the lymph capillaries through successively larger lymphatic vessels, referred to as lymphatic collecting vessels, until it is finally returned to the venous system through one of the two large ducts in the thoracic region. First is the right lymphatic duct, drains the lymph from the right arm and the right side of the head of the thorax. Next is the large thoracic duct, receives lymph from the rest of the body, and both ducts empty the lymph into the subclavian vein near your shoulder on their own side of the body. So the lymph nodes closely related to the immune system. They help protect the body by removing the foreign material such as bacteria and tumor cells from the lymphatic stream, and by producing lymphocytes that function the immune response. As lymph is transported toward the heart, it is filtered through the thousands of lymph nodes that cluster along the lymphatic vessels. Large clusters of lymph nodes are found in the first inguinal, singit, 2 axillary, then cervical regions. Within the lymph nodes are macrophages. Inside the lymph nodes, you have the macrophages. And what are these macrophages? They engulf and destroy bacteria, viruses, and other foreign substances in the lymph before it is returned to the blood. The lymphocytes is a type of white blood cells, respond to foreign substances in the lymphatic stream. And the lymph nodes, of course, vary in shape and size, but most are kidney shape, less than 1 inch, 2.5 cm long, and buried in the connective tissue that surrounds them. So each node is surrounded by a fibrous capsule from which strands called the trabeculae extend inward to divide the node into a number of compartments. So the parts of the lymph nodes, we have the cortex. The outer part of the node contains collections of lymphocytes called follicles. The follicles have dark staining centers called the germinal centers. These centers enlarge when lymphocytes are generating daughter cells called plasma cells, which release antibodies. The rest of the cortical cells are lymphocytes in transit that circulate continuously between the blood, lymph nodes, and lymphatic stream, performing their surveillance role. Then the other one part of the lymph node is the medulla, where phagocytic macrophages are located. This is afferent convex. The convex, it enters into afferent, this one. So this is convex, afferent, and then it exits here, efferent. So lymph enters the convex side of a lymph node through afferent lymphatic vessels. It then flows through a number of sinuses that cut through the lymph node and finally exits from the node at its intended region, the hilus, via efferent lymphatic vessels. Homeostatic imbalance. Lymph nodes help rid the body infectious agents and cancer cells, but they can also become the secondary cancer sites, particularly in cancers that use lymphatic vessels to spread throughout the body. Other lymphoid organs, the spleen, thymus gland, tonsils, and Peyer's patches of the intestine. So this is the anatomy of the lymphoid organs. I mean, these are the examples of the lymphoid organs. The spleen is a blood-rich organ that filters the blood. It is located on the left side of the abdominal cavity and extends to curl around the anterior aspect of the stomach. It filters and cleanses blood of bacteria, viruses, and other debris. The most important function is to destroy worn-out red blood cells and return some of their breakdown products to the liver. And the spleen stores platelets and acts as a blood reservoir. During hemorrhage, both the spleen and the liver contract and empty their contained blood into the circulation to help bring the blood volume back to normal levels. In the fetus, the spleen is an important hematopoietic or blood cell forming site, but only lymphocytes are produced by the adult spleen. This is an example of the spleen anatomy. You have there the trabeculate capsule, the pulp, the artery, and the vein. And you can find the spleen here, left side. Here is the left lobe of liver, then here is the stomach, then the spleen. The thymus gland, which functions at peak levels only during youth, so it functions only during our youthful years. It is a lymphatic mass found low in the throat, overlying the heart. It produces hormones, thymosin, and others that function in the programming of certain lymphocytes so they can carry out their protective roles in the body. So this is an example of thymus. You can find there the thymus low throat in the low, so the thymus gland, front view, and of course you can see there the medulla and the cortex. The tonsils are small masses of lymphatic tissue that ring the pharynx, the throat, where they found in the mucosa. Their job is to trap and remove any bacteria or other foreign pathogens entering the throat. They carry out this function efficiently that sometimes they become congested with bacteria and become red, swollen, and sore, which we call as tonsillitis. So this is the normal and this is the abnormal. Peyer's patches, which resemble tonsils, are found in the wall of the small intestine. It becomes part of the collection of small lymphoid tissues referred to as mucosa-associated lymphatic tissue or MALT, including the tonsils. MALT acts as a sentinel to protect the upper respiratory and digestive tracts from the never-ending attacks of foreign matter. Next is the body. To the body's defenders against these tiny but mighty enemies are two systems simply called the non-specific defense system. The non-specific defense system responds immediately to protect the body from all foreign substances, whatever they are provided by intact skin and mucous membranes, the inflammatory response, and a number of proteins produced by body cells. The non-specific defense system reduces the workload of the second protective arm, the specific defense system, by preventing entry and spread of microorganisms throughout the body. Most often, it refers to the mechanical barriers that cover body surfaces and to cells and chemicals that act on the initial battlefronts to protect the body from invading pathogens, harmful or disease-causing microorganisms. Fever is also considered to be a non-specific protective. Number 2 is a specific defense system, more commonly called the immune system. Mounts the attack against particular foreign substances. The immune system is a functional system rather than an organ system in an anatomical sense. Its structures are a variety of molecules and trillions of immune cells, which inhabit lymphatic tissues and circulate in the body cells. The most important of the immune cells are the immune systems. If this is operating effectively, it protects us from most bacteria, viruses, transplanted organs or grafts, and even our own cells that have turned against us. It attacks cells and indirectly by releasing mobilizing chemicals and protective antibody. This is the resulting highly specific resistance to disease. Non-specific body defense again refers to the mechanical barriers that cover body surfaces and to cells and chemicals that act on the initial battlefronts to protect the body from invading pathogens, harmful or disease-causing microorganisms. So for the non-specific body defenses, we have here first is the surface membrane barriers, which is the first line of defense. So first, under the first line of defense is the intact skin, particularly the epidermis, and under which, so this intact skin forms mechanical barrier that prevents entry of pathogens and other harmful substances into the body. Under which the intact skin, we have the acid mantle. Skin secretions make epidermal surface acidic, which inhibits bacterial growth. Sebum also contains bacterial killing chemicals. The keratin provides resistance against acids, alkalis, and bacterial enzymes. Number two, surface membrane barriers, first line of defense is the intact mucous membrane. Example of which, under which is the mucus that traps microorganisms in respiratory and digestive tracts. Nasal hairs filter and trap microorganisms in nasal passages. Cilia propel debris-laden mucus away from lower respiratory passages. Gastric juice contains concentrated hydrochloric acid and protein digesting enzymes that destroy pathogens in stomach. Acid mantle of vagina inhibits growth of bacteria and fungi in female reproductive tract, and lacrimal secretion or tears, saliva, continuously lubricate and cleanse eyes and oral cavity. Contain lysozyme, an enzyme that destroys microorganisms. Lysozymes are enzymes that destroy microorganisms. The second line of defense, cellular and chemical defenses. The example of which are the phagocytes, natural killer cells, the inflammatory response, and a variety of antimicrobial chemical substances that kill pathogens and help repair tissue, like the complement the entire. So the phagocytes engulf and destroy pathogens that breach surface membrane barriers organ. Examples are the macrophages or neutrophils, engulfs a foreign particle much the way an amoeba ingests a food particle, includes particle in a vacuole. The vacuole is then fused with the enzymatic contents of a lysozyme, and its contents are broken down or digested. Number two is the natural killer or NK cells, which police the body in blood and lymph, promotes the lysis by direct cell attack against a virus infected or cancerous body cells. They attack the target cell's membrane and release a lytic chemical called perforins. Shortly thereafter, the target cell's membranes and nucleus. And number three is the inflammatory response, is a non-specific response that is triggered whenever body tissues are injured, prevents spread of injurious agents to adjacent tissues, disposes of pathogens and dead tissue cells, and promotes tissue repair, releases chemical mediators that attract phagocytes to the area. So the four cardinal signs and major symptoms of an acute inflammation are redness, then heat, swelling, and pain. The inflammatory process begins with a chemical alarm. When the cells are injured, they release inflammatory chemicals, including histamine and kinins, that cause blood vessel in the involved area to dilate and capillaries to become leaky. Number two, it activates pain receptors and attract phagocytes and white blood cells to the area. So the latter phenomenon is called, the number three phenomenon is called chemotaxis, because the cells are following a chemical gradient. For the inflammatory response, there are three responses. First is it prevents the spread damaging agents to nearby tissues. 2, disposes of cell debris and pathogens. And 3, sets the stage for repair. Within an hour or so after the inflammatory process has begun, neutrophils are squeezing through the capillary walls, a process called diapedesis. Then monocytes follow neutrophils. Then these monocytes actually pour, but within 8 to 12 hours they become macrophages with insatiable appetite. The third line of defense are the immune response mediated by lymphocytes. So this third line of defense, its immune response is mediated by lymphocytes. Both protective antibodies and T cells invade the area to act specifically and directly against the damage. Pus is a mixture of dead or dying neutrophils, broken down tissue cells, and living and dead pathogens. Nana in our term. Nana. So if the inflammatory mechanisms fails to fully clear the area of debris, the sac of pus may become walled off, forming an abscess, hubag. Antimicrobial chemicals complement proteins and interferon, the body's most antimicrobial chemical apart from those produced in the inflammatory reaction. First is the complement. It refers to a group of at least 20 plasma proteins that circulate in the blood in an inactive stage. Group of plasma proteins that lyses microorganisms, enhances phagocytosis by opsonization, and intensifies inflammatory response. So when we say opsonization, that is the foreign cells are made to be sticky, to be phagocytized easily. So the cells, those cells which are not part of us, cells which are foreign, they are made to sticky so that they can easily be digested. So antimicrobial chemicals, again, when it becomes attached or fixed to foreign cells such as bacteria, fungi, or mismatched red blood cells, it is activated and becomes a major factor in the fight against the foreign cells. Complement fixation occurs when complement proteins bind to certain sugars or proteins such as antibodies on the foreign cell's surface. Example, the formation of membrane attack complexes or MAC that produce lesions complete with holes in the foreign cell surface. This allows water to rush into the cell, causing it to. Number 2 is the interferon, small proteins released by virus-infected cells that protect uninfected tissue cells from viral takeover, mobilize immune system. Fever is a systemic response to invading microorganisms. High body temperature inhibits multiplication of bacteria and enhances body repair processes. Systemic response is triggered by the pyrogens. When we say pyrogens, they are chemicals secreted by white blood cells and macrophages exposed to foreign cells or substances in the. High fevers are dangerous also. Bacteria require large amounts of iron and zinc to multiply. But during a fever, the liver and spleen gather up these nutrients, making them less available. In summary, to sum up, we have first line and second line of defense in non-specific defense mechanism. There are two lines of defense, first line, second line. In the first line, we have the skin, mucous membrane, secretions of skin and mucous membrane. In the second line, we have phagocytic cells, antimicrobial proteins, the inflammatory response. And in the third line of defense, this is now the specific defense mechanism or the immune system. We have the lymphocytes, antibodies, and macrophages. Let's go now to specific body response, or the third line of defense. The immune system's response to a threat, called the immune response, tremendously increases the inflammatory response, and it provides protection that is carefully targeted against specific antigens. The third line of defense, the important aspects of the immune response. So the specific body response is the third line of defense. Three important aspects of the immune response. First is it is antigen specific. It recognizes and acts against particular pathogens or foreign substances. It is systemic. Immunity is not restricted to the initial infection site. It's not only in that site, infection site, but it is all through the system. It is memory. It recognizes and mounts even stronger attacks on previously encountered pathogens. The two separate but overlapping arms of immunity were organized we have humoral immunity, also called antibody-mediated immunity, is provided by antibodies present in the body's humors or fluid. Cellular or cell-mediated immunity, when lymphocytes themselves defend the body because the protective factor is living cells. So the targets are, of course, the cancer cells, cells of foreign grafts, and virus. Antigens is any substance. Antigen is any substance capable of exciting our immune system and provoking an immune response. So most antigens are large, complex molecules that are normally present in our bodies, and they are foreign intruders or non-self. They are not in ourself, non-self. Example, foreign proteins, nucleic acids, many large carbohydrates, and some lipids. Proteins are the strongest antigen. Small molecules are not antigenic, but when they link up with our own proteins, the immune system may recognize the combination as foreign and mount an attack that is harmful rather than protective. That is called as, so there is allergy. So these troublesome molecules are called haptens or incomplete antigen. Chemicals that act as haptens are found in poison ivy, animal dander, some detergents, hair dyes, and so we go now to the cells of the. What are the cells of the immune system? We have the crucial cells of the immune system. We have the lymphocytes, exist in two major flavors. B lymphocytes or B cells produce antibodies and oversee humoral immunity. Humoral fluid. T lymphocytes, T cells are non-antibody producing lymphocytes that constitute the cell. And of course, we have the lymphocytes originate from hemocytoblasts and red bone marrow. The mature lymphocytes released from the marrow are essentially identical. Whether a given lymphocyte matures into a B cell or a T cell depends on where in the body it becomes immunocompetent, that is, capable of responding to a specific antigen by binding to it. B cells develop in bone marrow, T cells develops in lymphocytes. Once a lymphocyte is immunocompetent, it will be able to react to one distinct antigen and one only, because all the antigen receptors on its external surface are the same. Become immunocompetent before meeting antigens they may later attack. Once immunocompetent, T cells and B cells migrate to the lymph nodes and spleen where their encounters with antigens occur. So thus it is our genes, not antigens, that determine what specific foreign substances our immune system will be able to. Macrophages, they are the big eaters in the non-specific defense system, is to engulf foreign particles and rid them from the area. They present fragments of those antigens like signal flags on their own surfaces where they can be recognized by immunocompetent T cells. They act as antigen presenters in the specific defense system. Also secrete cytokine proteins called monokines that are important. To summarize, the immune system is a two-armed defensive system that uses lymphocytes, macrophages, and specific molecules to identify and destroy all substances, both living and non-living, that are in the body but are not recognized as being part of the body or as being self. So the immune system's ability of its cells first is to recognize foreign substances, like the antigens in the body, by binding to them, to communicate with one another so that the system as a whole mounts a response specific to body. Let's go now to the active and passive humoral. When we say active immunity, it can be naturally acquired or artificially acquired. This is very important. When your B cells encounter antigens and produce antibodies against them, that is active immunity. When your body B cells encounter antigens and produce antibodies against them, when they produce antibodies against that one, so it is called as active immunity. So as I said, we have naturally acquired. So when we say naturally acquired, during bacterial and viral infections, during which may develop the symptoms of the disease and suffer a little. So during infection, this naturally acquired immunity is developed. Then artificially required, from the word self-artificial, when we receive. Passive immunity is conferred naturally on a fetus when the mother's antibodies cross the placenta and enter the fetal circulation and after birth during breastfeeding. It is artificially conferred when one receives immune serum or gamma globulin. Gamma globulin is commonly administered after exposure to hepatitis. Other immune sera are used to treat poisonous snake bites, rabies. Vaccines contain dead or attenuated living but extremely weakened pathogens. We receive two benefits from vaccines. We are spared most of the signs and symptoms of the disease that would otherwise occur during the primary response, and the weakened antigens are still able to stimulate antibody production and promote immunological memory, so-called booster. Acquired immunity. Let's go now to acquired immunity. Acquired immunity, um, we have here active, naturally acquired. Example, when we say active and natural, that is infection. Example. But when we say active artificial, then that is vaccine or dead or attenuated pathogens. When we say passive but it's natural, then that is antibodies passed from mother to fetus via placenta or to infant in her milk. But when we say passive, artificial, then that is infection of immune serum or gamma globulin. Again, the two active, natural infection, active, artificial vaccine, passive natural antibodies or breast milk. Then passive, artificial is injection of immune serum. So the antibodies, also referred to as immunoglobulins or Igs, constitute the gamma globulin part of blood proteins, are soluble proteins secreted by cells or their plasma cell offspring in response to an antigen, and they are capable of binding specifically with that. So basic antibody structures for amino acid polypeptide chains linked together by disulfide sulfur to sulfur bonds. Two of the four chains are identical and contain approximately 400 amino acids each. These are the heavy chains. Then the other two chains, the light chains, are also identical to each other but are only about half as long. So they are shaped as T or Y. Each of the four chains forming antibody had a variable V region at one end and a much larger constant C region. Constant regions of the antibody chain serve common functions in all antibodies. They determine the type of antibody form, antibody class, as well as how the antibody class will carry out its immune roles in the body, and the cell types or chemicals with which the. So we have here the classes of antibodies. We have IgD, IgM, IgG, IgA, then IgE. So we have here, when we say Ig, they virtually always attach to B cells. Then IgM attached also to B cell. IgG, they are mostly found in plasma. Then, of course, in the circulating antibodies. And IgA can be found in the plasma also, saliva, tears, intestinal juices, and milk. And IgE can be found also in plasma, mucosa of the GIT and respiratory tracts. IgM can fix complement, and IgG can cross the placental barrier, provides passive immunity to fetus, and fixes complement. And IgA can be found mainly in mucus, and IgA also involved in allergies. So that's. So what are the functions of cells and molecules involved in immunity? Again, B cell, it is a lymphocyte that resides in the lymph nodes, spleen, or other lymphoid tissue where it is induced to replicate by antigen binding and help T cell and helper T cell interactions. Its progeny clone members form plasma. So plasma cells are antibody producing machine producing huge numbers of the same antibody. It represents further specialization of B cell clone. And helper T cell, a regulatory T cell that binds with a specific antigen presented by a macrophage. It simulates the production of other immune cells, cytotoxic cells, T cells, and B cells to help fight the invader, acts both directly and indirectly by. Cytotoxic T cell, also called a killer T cell, activity enhanced by helper T cells. It especially is killing virus. It especially is killing virus invaded body cells as well as body cells that have become cancerous, involved in graft. And the suppressor T cells slows or stops the activity of B and T cells once infection or attack by foreign cells has been. And memory cell, descendants of an activated B cell or T cell generated during the initial immune response, may exist in the body for years thereafter, enabling it to respond quickly and efficiently to subsequent infections or meetings with. Macrophage engulfs and digests antigens that it encounters and presents parts of them in plasma membrane for recognition by T cells. So the molecules, let's go to the molecules. Antibody or immunoglobulin, protein produced by a B cell or its plasma cell offspring and released into the body fluids in the blood, lymph, saliva, mucus, etc., where it attaches to antigens, causing neutralization, precipitation, or agglutination, which marks the antigens for destruction by phagocytes or complement. So the lymphokines, they are cytokine chemicals released by sensitized T cells. Macrophage migration inhibiting factor MIF inhibits macrophage migration and keeps them in local area. Interleukin 2 stimulates T cells and B cells to proliferate. And helper factors enhance antibody formation by. Suppressor factors suppress antibody formation or T cell mediated immune response, and chemotactic factors attract leukocytes like the neutrophils, eosinophils, and basophils into inflamed area. And then the perforin is a cell toxin, causes cell lysis. Gamma interferon helps make tissue cells resistant to viral infection, activates macrophages, activates NK cells, enhances maturation. Monokines are cytokines chemicals released by activated macrophages. The interleukin 1 stimulates T cells to proliferate and cause fever. And the tumor necrosis factor, like perforin, causes cell killing, attracts granulocytes, activates T cells and macrophages. And the complement, the group of blood-borne proteins activated after binding to antibody-covered antigens, when activated, complement causes lysis of the microorganism and enhances inflammatory response. For the antigen, substance capable of provoking an immune response, typically a large complex molecule not normally. Organ transplants and rejection. 4 major types of grafts. We have autografts, isograft, allograft, and xenografts. For autografts, they are tissue grafts transplanted from one site to another in the same person. Isograft are tissue grafts donated by a genetically identical person, like identical twin. Allografts are tissue grafts taken from an unrelated person. Xenografts are tissue grafts harvested from a different animal species, such as transplanting a baboon heart into a human. Immunosuppressive therapy. They are given during surgery to prevent rejection, like the corticosteroid to suppress inflammation, cytotoxic drugs, radiation therapy, and the most important disorders of the immune system are allergies, immunodeficiencies, and autoimmune disease. When we say allergies, hypersensitivities, they are abnormally vigorous immune damage as it fights off a perceived threat that would otherwise be harmless to the body. The most common type are the immediate hypersensitivity, also called acute hypersensitivity, is triggered by the release of a flood of histamine when IgE antibodies bind to. So histamine causes small blood vessels in the area to become dilated and leaky, and is largely to blame for the best recognized symptoms of allergy, like the runny nose, watery eyes, itching, reddened skin. And over-the-counter meds are the antihistamine. Anaphylactic shock, the body-wide or systemic acute allergic reaction, occurs when the allergen directly enters the blood and circulates rapidly through the body, as might happen with certain bee stain. Immunodeficiencies include both congenital and acquired conditions in which the production or function of immune cells or complement is abnormal. Most of the devastating AIDS-acquired immune deficiency syndrome cripples the immune system by interfering with the activity of helper. Autoimmune diseases are when the immune system loses its ability. So what do we mean by autoimmune diseases? Our immune system loses its ability to distinguish friend from foe. That is to tolerate self-antigens while still recognizing and attacking foreign antigens. The body produces antibodies, autoantibodies, and sensitized T cells that attack, and damage its own tissues. So it is one's immune system that produces the disorder. So the most common autoimmune diseases are multiple sclerosis, which destroys the white matter or myelin sheaths of the brain and spinal cord. We have myasthenia gravis, which impairs communication between nerves and skeletal muscle. Graves' disease, in which the thyroid gland produces excessive amounts of thyroxine. And type 1 diabetes mellitus, which destroys pancreatic beta cells, resulting in deficient production of insulin. The systemic lupus erythematosus also, systemic disease that occurs mainly in young females and particularly affects the kidneys, hearts, lungs, and skin. Glomerulonephritis, a severe impairment of kidney function, and rheumatoid arthritis, which systematically destroys